Good morning everyone and welcome to this 4D BISBA webinar dedicated to omic sciences. My name is Lindsay Periga from Toulouse Biotechnology Institute at INSA and I'm working as application engineer on the metabolomic and fluxomic platform MetaTool. Today I will present you some results obtained within industrial biotechnology project and concerning the improvement of riboflavin bioproduction using C14 isotopic profiling strategies. This work was done thanks to a Franco-German collaboration between the MetaTool platform, the University of Saarland with the team of Professor Wittmann, and the BASF company, and led to a publication in Metabolic Engineering in 2018. First, some words about the global context of this study. So the riboflavin, also known as vitamin B2, is currently produced by plant or microorganism. But it is an essential vitamin for humans and animals. It is the precursor of flavin adenine dinucleotide and flavin mononucleotide, and the riboflavin is mainly used as food and feed additive or food colorant. It's a yellow pigment. In terms of economy, it is an increasing market, 9,000 tons produced in 2015 against 4,000 tons in 2009. Chemical processes linked to fossil fuels initially produced this molecule, but now it is an exclusive biotechnology production, and the main organism used and operated by the BASF company in South Korea is the filamentous fungus H. with the production yields around 20 grams per liter. So, as you can see on this slide, the bioproduction of riboflavin is a multi-compartmented process, and the main growth sources are vegetable oil. Also, each carbon with the different color, gray, red and blue, coming from different metabolic pathway with some uncertainties. For industrial bioproduction, not only vegetable oil is used, but also, and in addition, yeast extract, peptone and corn steep liquor. Therefore, it is really complex outside and inside of the cells. So, to keep up with the market demand, it will be benefit to better understand the riboflavin production by H-biagocp in order to propose a new strategy for cultivation, and it is the purpose of this work. So, most of the carbon contribution are well described for the riboflavin biosynthesis, but there is still some uncertainty. Indeed, regarding the origin of the blue glycine-derived carbon, serine and or glycine might contribute to those carbon. And regarding the origin of the red C1-derived carbon, serine, glycine and formate, which are all present into the medium, might contribute to this carbon. So, to answer those questions and to establish if yes or not those nutrients contribute to the formation of the carbon skeleton of the riboflavin, we decided to use C14 labeling experiment strategies with labeled glycine, serine or formate, and made NMR analysis complementary to mass spectrometry experiment carried out in Germany. And NMR will provide us positional information really useful to this project. Regarding the cultivation part, it is a complex process that starts first with the cultivation on solid medium to slowly wake up the cells during 5 to 7 days. Then we can perform a liquid preculture on yeast extract and rapeseed oil during 40 hours, and finally the main culture made on rich medium complemented with amino acid during 144 hours to reach a production yield of 20 grams per liter. It is during this third step that labeled glycine, serine and formate will be introduced into the medium in order to perform the C14 labeling experiment. The first work done was to fully characterize the C14 NMR spectrum of riboflavin. To do that, we work on purified compound produced thanks to an unlabeled culture. And we have to know that the riboflavin at high concentration is not soluble in water. so it was really easy to separate it from the medium, and we will be able to dissolve this purified compound into deuterated dimethyl sulfoxide, compatible with NMR analysis, and recorded in the spectrum on 800 MHz. Then we attribute each signal to one carbon of the molecule, and for the rest of the study, we will focus our analysis on the carbon 2, the carbon 4 alpha, and the carbon 10 alpha between 130 and 160 ppm. Our collaborator in Germany performed cultivation on C13 glycine, C13 serine, and C13 format separately, and thanks to C13 NMR, we were able to measure the positional C13 enrichment for the carbon 2, the carbon 4 alpha, and the carbon 10 alpha. And we can notice here that the free compound contributes to the construction of the carbon skeleton of riboflavin, formatted serine for the C2, and glycine for the C4 alpha and the C10 alpha. Those results were confirmed with mass spectrometry analysis with M+1 and M+2 species present in the analysis. So, for each type of label experiment, we measure quantitatively the C13 enrichment to evaluate the contribution of each substrate to each carbon, and the values are reported into this table. Luckily, we well identify the potential candidates at the beginning of the project as carbon donor for the C2, C4 alpha, and C10 alpha, like glycine, serine, and formate. Okay, when we have a look on glycine experiment, into the medium we could have unlabeled carbon sources coming from vegetable oil or yeast extract, leading to no C13 enrichment, and labeled glycine added fully, labeled glycine, or labeled glycine on position 2, leading potentially to 3 cases. First, labeled C10 alpha and C4 alpha, or labeled C2, C4 alpha and C10 alpha, or only labeled C2 and C4 alpha. That's theoretically what could happen. Experimentally, we observe labeling incorporation into C10 alpha and C4 alpha around 83%. When we have a look on serine experiments, again in the medium we could have unlabeled carbon sources, leading to no C13 enrichment, and labeled serine added on position 3, leading to a C2 enrichment. And what we can observe experimentally is an enrichment of the C2 around 11%. Finally, when we have a look on format experiment, we again could have unlabeled carbon sources with no C13 enrichment, and fully labeled format, which is a C1 compound, leading to C2 enrichment. And what we can observe experimentally, it's an enrichment on the C2 around 23%. All this enrichment information were used in combination with a mass spectrometry data into a flux calculation software used by our collaborator in Germany, and now we can properly show the contribution of each nutrient to the constitution of the carbon skeleton of riboflavin. We can conclude that glycine and serine contribute to the formation of the C4 alpha and C10 alpha of riboflavin. It's the picture A. And formate, serine contribute to the formation of the C2 of the riboflavin, which is the picture B. To summarize, thanks to C13 profiling, isotopic profiling, we can describe the complete carbon contribution for riboflavin production. And we will be able now to propose an improvement of feeding medium in order to increase the production yield of riboflavin. Indeed, with the supplementation of the medium with format only with we increase up to 140% the production yield during the 12 first hour. Nevertheless, if we add serine and formate into the medium, We maintain this production yield up to 24 hours, which is really promising. To finish, I would like to thank all the collaborators of this project from the Metatool platform, the Salon University, and the BASF company, and of course you for your attention. Thank you.